I am Malofi. In this video, I will present my final year project which entitled Toxicity Study of Cold Nanoparticles using Zebrafish. Okay, so the outline of this presentation will be introduction, objective, methodology, results and discussion, conclusion and future work. Okay, so for the introduction, gold nanoparticles are commercially used in both industrial and biomedical applications. Due to its unique properties in nanometer size, gold nanoparticles are highly remarkable in the biomedical application as it was used as drug carriers, chemicals and biological sensing, X-ray imaging, and more. Even though the properties of gold is inert, the removal rate of gold from the circulation and tissues is relatively low. This may be due to its nanoparticle size that make it easily absorbed in cells or tissues. As a result, this could lead to health problems if the gold nanoparticles were continually consumed by the human. This has induced question whether the gold nanoparticles is toxic to human in the long term. So, to study the potential toxicity of gold nanoparticles, zebrafish is used because it has fast development, high fecundity, transparent embryo, and possesses a resemblance of the human genome. This research is done to explore how the gold nanoparticles can affect the development of zebrafish by varying the concentration of the gold solution. Okay, so there are two objectives of my research work, which is first to synthesize and characterize gold nanoparticles produced at different concentration of reducing agent, and second to study the toxicity of gold nanoparticles produced at different concentration using zebrafish. Okay, so the overall methodology of my research can be divided into two parts. The first part is to synthesis gold nanoparticles and the characterization. Second is to introduce gold nanoparticles to zebrafish embryo and observation on zebrafish. And lastly is to conclude. Okay, so for the result and discussion, I will start with the synthesis result. For the synthesis of gold nanoparticles, the method used was Turkevish method from Turkevish et al. In this method, trisodium citrate was used as reducing agent to reduce gold ion to gold particle. The concentration of reducing agent was manipulated to get different size of gold nanoparticles, where the concentration of reducing agent used is 0.5%, 3%, and 4%, while the concentration of tetrachloroauric acid was kept constant at 0.01 molarity. In the beginning, the color of tetrachloroauric acid was pale yellow as you can see in this picture, and after reducing agent was added, the color immediately changed to colorless. After the addition of the reducing agent, the solution were heated until the color turns to wine red or dark purple. This color change indicates that the gold nanoparticles have occurred in the solution. And the solution was labeled as sample 1, sample 2, and sample 3 according to the concentration of reducing agent, which is 0.5%, 3%, and 4%. From the observation, we can see that the color of the final solution was darker at higher concentration of trisodium citrate. Okay, so for the characterization of gold nanoparticles, three different techniques was used, which is UV-Vis, PSEM, and EDS. After the synthesis of gold nanoparticles, UV-Vis was done to detect the presence of gold nanoparticles in the sample. Then PSEM analysis was done for the morphological analysis of the gold nanoparticles. This is to check the size or shapes of gold nanoparticles obtained in the sample. And lastly, we do the EDS analysis on the chosen sample, which will be used in the toxicity analysis part to obtain the elemental composition of the sample. Good. Okay, so this is the result obtained from the UV-Vis analysis. Firstly, we can see that the peak of all three samples appeared at the wavelength in the range of 522 to 524 nanometer, which correspond to that reported by Martinez et al., where gold nanoparticle spectra for spherical shaped gold will be between 520 and 560 nanometer. And the wavelength, as we can see, the wavelength of the sample is different, which shows that the size of gold nanoparticle produced will be different when the different concentration of reducing agent was used during synthesis. For the sample 3, the wavelength was seen broader than other sample, which shows that the agglomeration of nanoparticles might have occurred in the sample. Moving on to the PCM analysis, this is the result for the PCM analysis, result obtained from the PCM analysis. In this table, we can conclude that the average diameter of gold nanoparticles obtained at 0.58% was approximately 20 nanometer, and 30 nanometer for 3 weight percent and 15 nanometer for 4 weight percent. Okay, so from the result, the result shows that the diameter of gold nanoparticles increased with an increase in trisodium citrate concentration. This is due to the agglomeration of gold nanoparticles that occur prior to the PCM analysis of the sample. Smaller particle tends to have higher mobility, and thus the collision between the particles were higher. This will cause the agglomeration occurs easily and cause the size of gold nanoparticles seen larger under the microscope. Report from Ahmed Tabrizi and Fatma Ayhan shows that such gold nanoparticles was larger than it's supposed to be at certain ratios of trisodium citrate to gold. And another reason was this, because as the concentration of trisodium citrate increased, the reducing agent will present in excess in the solution, which will lead to over-reduction of gold. Okay, so this from the EDS, uh, for the EDS, sample 1 is chosen to undergo EDS analysis. This is because from the PCM analysis, sample 1 turns out to have the smallest size of gold nanoparticles. Thus, sample 1 is the most suitable to be used in toxicity analysis. From the results, we can see that carbon, oxygen, natrium, chlor, chlorine, aluminum, and gold is present in the sample. And as we can see here, aluminum has the highest peak amongst the other elements with the weight percent of 23.69 weight percent. This is because the stack used to place the sample itself was mainly made of aluminum, while the percentage of gold present in the sample 1 is 21.90%, which clearly prove that the gold is present in the sample. The other elements, carbon, oxygen, and natrium, are also present in the sample, which coming from the unreacted reagent from the synthesis process. Okay, moving on to toxicity analysis of gold nanoparticles. Okay, at 24 hours, only from the survival rate of zebrafish, at first 24 hours, only one embryo that does not survive, which is at the concentration of 15.63 microgram per mil. This is concluded as error because the embryo was not at the maximum concentration of gold nanoparticles. And this is also because due to injury during the transferring process of embryo from petri dish to the sample plate. Okay, so at 120 hours, the trend of the survival rate of zebrafish are increasing from 1000 to 125 microgram per mil, as you can see here, which prove that higher concentration of good nanoparticles poses more toxicity to the zebrafish. And however, the trend of survival rate for the concentration at 62.5 to 15.63 microgram per mil fluctuates. This is might due to contamination of the media. And for the control group, all embryo are survived. Okay, for the mortality rate, the exposure to gold nanoparticles will result in increased mortality rate when the concentration of gold nanoparticles arise from 15 to 1000 microgram per mil. The mortality rate of the zebrafish was the highest at 1000 microgram per mil with a value of 33.33% and 0 at 135 microgram per mil. Even though the mortality rate at uh, not 15.60 but 31.25 microgram per mil was quite high at 16.67%. It can be concluded due to error during the preparation of the sample. Okay, for the hatching rate, one of the most important events to understand the nanotoxicity of gold is the zebrafish hatching. The hatching period starts as you can see here, the hatching period starts at 48 hours and at 72 hours all embryos have hatched. And the hatching rate for the control group is the highest at 48 hours with 50%, while for 1000 microgram per mil concentration, the hatching rate is only 8.33%. This shows that the development of zebrafish organ was lower in the medium that contained high concentration of gold nanoparticles than in a controlled medium. Okay, after the exposure of the sample for 5 days, the embryo larva shows almost no abnormalities except for mud scoliosis. Scoliosis is the abnormalities of the embryo's trunk, and these abnormalities prove that gold nanoparticles do cause toxicity in the development of zebrafish. Okay, for the conclusion, throughout the research, it can be concluded that gold nanoparticles do possess toxicity behavior when exposed to zebrafish. The gold nanoparticles concentration of 1000 microgram per mil was shown to be the most toxic and cause abnormalities on the zebrafish organ development. Mud scoliosis can be observed on the zebrafish larva at the concentration of 1000 microgram per mil after 132 hours of exposure to the samples. Aside from decreased survival rate and increased mortality rate, the hatching rate of the embryo also reduced as the concentration of gold nanoparticles in the sample increased. For the synthesis of gold nanoparticles, trisodium citrate plays an important role as both reducing and capping agent. The Zebrafish method can be used to produce spherical-shaped gold nanoparticles with the size range of 70 to 22 nanometer depending on the concentration of the reducing agent. And lastly, a higher concentration of trisodium citrate will result in larger size of gold nanoparticles. Okay, for the recommendation or future work, there are there are various methods that can be used to synthesize gold nanoparticles aside from the Zebrafish method, which are brass method, seeded growth method, and etc. This method will give a wide range of size and shape of gold nanoparticles. For the Zebrafish method, other for the Zebrafish method, other manipulating variables can be done to see the effect on gold nanoparticle size and shapes. This includes using different types of reducing agent, varying pH, temperature of solution, capping agent, and etc. And for the characterization, other techniques as SEM, TEM, and FTIR and more will also can be used, which will give more variety of result. And lastly, for the toxicity study of gold nanoparticles, the toxicity study of gold nanoparticles can be further explored by using samples with different size, shape, and shapes to verify the other factors that makes the gold nanoparticles toxic to humans. Okay, so that's all from me. Thank you for listening.